Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. In the heart of Cambridge, England, during the early 1950s, a revolution was brewing in the most unlikely of places, a cramped laboratory where scientists were wrestling with one of biology's greatest mysteries. What carried the instructions? That made a rose red, a human tall or a bacterium resistant to disease. For centuries, the mechanism of heredity had remained stubbornly hidden, locked within the microscopic world of cells. The story begins decades earlier, when Swiss physician Friedrich Miescher first isolated a phosphorus-rich substance from white blood cell nuclei in 1869. He called it nuclein, though he had no idea he had discovered what would later be known as deoxyribonucleic acid, or DNA. At the time, most scientists believed proteins carried hereditary information. They seemed complex enough for such an important job. DNA, with its simple four-letter chemical alphabet, appeared too mundane to house life's blueprint. By the 1920s, British scientist Frederick Griffith conducted experiments with pneumonia bacteria that would change everything. He discovered that dead disease-causing bacteria could somehow transform living, harmless bacteria into killers. Something was being transferred between them, a transforming principle that carried genetic instructions. This phenomenon suggested that hereditary material was not just theoretical, but actively transferable between organisms. The transforming principle remained a mystery until 1944, when Oswald Avery, Colin MacLeod, and Macklin McCarty at Rockefeller University proved definitively that DNA, not protein, was the hereditary material. Their meticulous experiments showed that when they destroyed proteins or RNA in their bacterial extracts, transformation still occurred. But when they destroyed DNA, transformation stopped completely. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free and it helps us grow this community together. This revelation set off an international race to understand DNA's structure. Scientists worldwide recognized that whoever cracked DNA's three-dimensional architecture would unlock the secret of how genetic information was stored, copied, and passed from generation to generation. The stakes were enormous. This knowledge could revolutionize medicine, agriculture, and our fundamental understanding of life itself. Several research groups emerged as frontrunners in this scientific competition. At King's College London, Maurice Wilkins and Rosalind Franklin were using X-ray crystallography to probe DNA structure. Meanwhile, at Cambridge University, James Watson and Francis Crick were building three-dimensional models, trying to piece together the molecular puzzle from available chemical and physical evidence. The challenge was formidable. DNA molecules were incredibly tiny, far too small to see even with the most powerful microscopes of the era. Scientists had to rely on indirect methods, interpreting shadows and patterns created when X-rays bounced off DNA crystals. It was like trying to understand the architecture of a building by studying only the shadows it cast on the ground. Chemical analysis had revealed DNA's basic components, 4 bases called adenine, thymine, guanine, and cytosine, linked to a sugar phosphate backbone. But how were these components arranged in three-dimensional space? Was DNA a single strand or multiple strands? How did it replicate itself? These questions drove researchers to work 18-hour days, fueled by the knowledge that scientific immortality awaited whoever solved the puzzle first. The race was about to intensify dramatically, with personalities, egos, and brilliant insights colliding in ways that would forever change our understanding of life's most fundamental processes. In a basement laboratory at King's College London, Rosalind Franklin bent over her X-ray crystallography equipment, adjusting the angle of a tiny DNA fiber with microscopic precision. The year was 1951, and Franklin was about to create one of the most important photographs in scientific history, though she wouldn't know it at the time. X-ray crystallography was the cutting-edge technique for probing molecular structure, but it required extraordinary skill and patience. Franklin had to purify DNA, form it into perfect crystals, and then bombard these crystals with precisely focused X-rays. When the X-rays encountered DNA's atoms, they scattered in specific patterns that could reveal the molecule's three-dimensional architecture. Reading these patterns required both mathematical expertise and intuitive insight. Franklin's background made her uniquely qualified for this challenge. She had earned her doctorate in physical chemistry from Cambridge University, then spent 4 years in Paris learning advanced X-ray techniques from Jacques Meringue, one of Europe's leading crystallographers. She understood how molecular structure influenced X-ray diffraction patterns better than almost anyone in Britain. Working with graduate student Raymond Gosling, Franklin discovered that DNA could exist in two distinct forms depending on humidity levels. In dry conditions, it formed what she called the A form, a compact crystalline structure. But when humidity increased above 75%, DNA transformed into the B form, longer, more hydrated, and closer to its natural state. Inside living cells, the B form proved crucial because it produced clearer, more interpretable X-ray diffraction patterns. Franklin's most famous photograph, known as Photo 51, captured the B form's distinctive pattern. The image showed a clear X-shaped pattern in the center, surrounded by diamond-shaped arrangements of spots. To trained eyes, these patterns revealed fundamental information about DNA's structure. From Photo 51 and hundreds of similar images, Franklin deduced several critical facts. The X-shaped pattern indicated that DNA was helical, twisted like a spiral staircase. The spacing between the diffraction spots revealed that the helix made a complete turn every 34 angstroms, with bases stacked 3.4 angstroms apart. This meant there were 10 bases per complete turn of the helix. Franklin's measurements also revealed that DNA consisted of 2 strands running in opposite directions, with phosphate groups on the outside and bases on the inside. The uniform diameter suggested that larger bases always paired with smaller ones, specifically adenine with thymine and guanine with cytosine. This complementary base pairing would prove essential for understanding how DNA replicated itself. But Franklin was cautious about drawing conclusions too quickly. She wanted overwhelming evidence before proposing a specific structural model. Her scientific training emphasized careful data collection and rigorous analysis over hasty theorizing. This methodical approach would ultimately cost her priority in the discovery race. Meanwhile, Franklin's colleague, Maurice Wilkins, had a complicated relationship with her work. As the senior researcher who had initially recruited Franklin to the DNA project, Wilkins sometimes felt sidelined by her expertise and dedication. Tensions arose over data sharing and collaboration, creating an atmosphere of competition rather than cooperation within their own laboratory. Unknown to Franklin, her X-ray data was being shared beyond King's College. Wilkins showed Photo 50, one to James Watson, during a visit to Cambridge University in January 1953. Watson immediately recognized the image's significance. It provided the missing pieces needed to build an accurate model of DNA's structure. Franklin's meticulous measurements had revealed DNA's helical nature, its dimensions, and the arrangement of its chemical components. Her work represented months of 18-hour days, countless failed experiments, and mathematical calculations that filled notebook after notebook. She had essentially solved DNA's structure through pure experimental science, yet Franklin remained unaware that her data was fueling model-building efforts at Cambridge University, where Watson and Crick were racing to beat Linus Pauling, the world's most famous structural chemist, to the ultimate prize in molecular biology. 23 miles northeast of London, in Cambridge University's Cavendish Laboratory, 2 scientists were taking a radically different approach to solving DNA's structure. James Watson, a 24-year-old American biologist, and Francis Crick, a 35-year-old British physicist, believed that building physical models could reveal molecular architecture faster than collecting endless experimental data. Their collaboration began in October 1951, when Watson arrived at Cambridge as a postdoctoral researcher. Crick was working toward his doctorate, studying protein structure using X-ray crystallography. Both men shared an obsession with understanding how genetic information was stored and transmitted, and they quickly realized their complementary skills could crack DNA's code. Watson brought deep knowledge of genetics and biochemistry, understanding how genes functioned in living organisms. Crick contributed expertise in physics and structural analysis, knowing how molecular shapes determined biological function. Together they represented a new breed of scientists, interdisciplinary researchers, who could bridge the gap between physics, chemistry, and biology. Their model-building approach was both revolutionary and risky. Instead of spending years collecting X-ray data like their competitors, Watson and Crick gathered existing information from scientific literature and conference presentations, then built three-dimensional models using metal rods, plates, and clamps. If their models satisfied all known chemical and physical constraints, they reasoned, the correct structure would emerge. Their first attempt in November 1951 was spectacular failure. Watson and Crick proposed a three-strand model with phosphate groups in the center and bases pointing outward. They invited Franklin and Wilkins to Cambridge to evaluate their model, confident they had solved DNA's structure. Franklin demolished their proposal in minutes. She pointed out that their model violated basic chemistry. The phosphate groups couldn't be held together by hydrogen bonds in the way Watson and Crick had proposed. Moreover, their model didn't match her X-ray data, which clearly showed phosphates on the outside of the molecule. The humiliation was complete, and Lawrence Bragg, head of the Cavendish Laboratory, temporarily banned Watson and Crick from working on DNA, but the ban couldn't suppress their curiosity. Watson and Crick continued thinking about DNA structure, discussing it during long walks and coffee breaks. They realized their first model failed because they hadn't fully understood the chemical constraints governing molecular structure. They needed better data and deeper insight into how DNA's components fit together. Their breakthrough came when they learned about Chargaff's rules, chemical analyses showing that DNA always contained equal amounts of adenine and thymine and equal amounts of guanine and cytosine. This one-to-one ratio suggested that these bases paired specifically with each other, but Watson and Crick initially couldn't figure out how this pairing worked. The competition intensified dramatically in late 1952 when Linus Pauling, winner of the Nobel Prize in Chemistry and the world's leading authority on molecular structure, entered the race. Pauling had already determined the structure of proteins using model-building techniques similar to Watson and Crick's approach. If anyone could solve DNA's structure through pure theoretical analysis, it was Pauling. In February 1953, Pauling published a paper proposing a three-strand DNA model with phosphate groups in the center, essentially the same incorrect structure Watson and Crick had proposed 15 months earlier. But Pauling's reputation was so formidable that many scientists initially accepted his model, despite its chemical impossibilities. Watson and Crick realized this was their chance. Pauling's error bought them time to build a correct model before the master corrected his mistake. They knew Pauling would eventually recognize his error and propose the right structure, probably within weeks or months. The pressure was enormous. Watson and Crick had to synthesize Franklin's X-ray data, Chargaff's base pairing rules, and their own structural insights into a single, coherent model. They worked frantically, building and rebuilding molecular models, testing different configurations against known chemical and physical constraints. The race was entering its final, most intense phase. The morning of February 28, 1953, found James Watson in his Cambridge laboratory, manipulating cardboard cutouts of DNA's chemical bases. For weeks, he had been struggling with a fundamental problem. How did adenine, thymine, guanine, and cytosine fit together inside the DNA molecule? The answer would determine whether their model-building approach could succeed. Watson had been assuming that identical bases paired with each other, adenine with adenine, thymine with thymine, but this arrangement produced irregular shapes that couldn't fit inside a uniform helical structure. Frustrated and running out of ideas, he decided to try a different approach suggested by Jerry Donohue, an American chemist working in the same laboratory. Donohue had pointed out that Watson was using the wrong chemical forms of the bases. Textbooks showed adenine and guanine in their enol forms, but Donohue insisted these bases actually existed in keto forms under normal cellular conditions. This seemingly minor correction would prove revolutionary. When Watson made cardboard cutouts representing the correct keto forms, everything changed. He discovered that adenine paired perfectly with thymine, and guanine paired perfectly with cytosine. Both base pairs had identical shapes and dimensions. They were complementary. This meant that regardless of which bases occurred at any position, in the DNA sequence, the overall width of the molecule remained constant. The implications hit Watson like a thunderbolt. If adenine always paired with thymine and guanine always paired with cytosine, then the two strands of DNA were complementary, copies of each other. Each strand contained all the information needed to reconstruct its partner. This was the copying mechanism that had eluded scientists for decades. Watson immediately called Francis Crick, who was having lunch at a nearby restaurant. When Crick arrived at the laboratory, Watson showed him the perfectly matching base pairs. Crick grasped the significance instantly. They had discovered not just DNA's structure, but also how it replicated itself. The complementary base pairing meant that each strand could serve as a template for building a new partner strand. Working with fevered intensity, Watson and Crick spent the next 3 weeks building their final model. They used metal rods and plates to construct two intertwined helical strands with bases paired in the center, like rungs on a twisted ladder. The phosphate sugar backbones formed the outer rails, exactly as Franklin's X-ray data had indicated. Their model satisfied every known constraint. The helix made one complete turn every 34 angstroms, with 10 base pairs per turn, matching Franklin's measurements precisely. The anti-parallel arrangement of strands, running in opposite directions, explained the X-ray diffraction patterns. The complementary base pairing accounted for Chargaff's 1-to-1 ratios, but Watson and Crick needed independent verification. They invited colleagues to examine their model, looking for flaws or inconsistencies. Organic chemist Alexander Todd confirmed that their proposed chemical bonds were realistic. Crystallographer William Asbury agreed that the structure matched available X-ray data. Most importantly, Maurice Wilkins brought Franklin's latest measurements from King's College. Her data fit their model perfectly. Every angle, every distance, every structural feature corresponded to her experimental observations. The double helix wasn't just theoretically elegant, it was experimentally accurate. The final test came when they calculated what X-ray diffraction pattern their model should produce, then compared it to Franklin's actual photographs. The match was extraordinary. Their theoretical model predicted precisely the patterns Franklin had observed, including the distinctive X shape in photo 51 and the detailed spacing of diffraction spots. On March 7, 1953, Watson and Crick invited visitors to their laboratory to see the completed model. Standing nearly 6 feet tall, the double helix twisted gracefully upward, its metal components gleaming under laboratory lights. The structure was breathtakingly beautiful, an elegant spiral staircase encoding the secrets of life itself. Francis Crick couldn't contain his excitement. That lunchtime at the Eagle pub, he announced to anyone who would listen that he and Watson had found the secret of life. While this claim seemed grandiose, it would prove remarkably prescient. The double Helix model would revolutionize biology, medicine, and human understanding of heredity. The beauty of Watson and Crick's double helix model wasn't just aesthetic, it was functional. Within hours of completing their structure, both scientists realized they had discovered something far more profound than DNA's architecture. They had uncovered the molecular mechanism that allowed genetic information to be copied with extraordinary fidelity from one generation to the next. The key insight lay in the complementary base pairing. Since adenine always bonded with thymine and guanine always bonded with cytosine, each strand of the double helix contained all the information needed to reconstruct its partner. If the two strands separated, each could serve as a template for building a new complementary strand. The result would be two identical double helices, each containing one original strand and one newly synthesized strand. This semi-conservative replication mechanism solved a puzzle that had mystified biologists for decades. How could cells make perfect copies of their genetic material before dividing. How could hereditary information be transmitted accurately from parents to offspring? The double helix provided an elegant answer. DNA replicated itself by unzipping and using each strand as a blueprint for constructing its complement. Watson and Crick envisioned the replication process as a molecular dance of extraordinary precision. Enzymes would unwind the double helix, separating the two strands like opening a zipper. Other enzymes would then travel along each single strand, reading the sequence of bases and adding complementary bases to construct new partner strands. Where the original strand had adenine, the enzyme would add thymine. Where it had guanine, the enzyme would add cytosine. The implications were staggering. This mechanism explained how a single fertilized egg could divide repeatedly while maintaining identical genetic information in every cell. It revealed how genetic mutations occurred, occasional errors during replication that changed the base sequence. It suggested how evolution worked, random changes in DNA sequence that could be inherited if they provided survival advantages. But the double helix model also raised profound new questions. What enzymes actually performed DNA replication? How fast did the process occur? How were errors corrected? How was the enormous length of DNA in human cells, nearly 6 billion base pairs, copied accurately during each cell division? Watson and Crick realized that answering these questions would require decades of additional research. Their model provided the foundation, but understanding the complete machinery of genetic replication would demand the efforts of thousands of scientists worldwide. They had opened a door to an entirely new field of biology. The replication mechanism also suggested how genetic information was organized and expressed. If DNA stored hereditary instructions in its base sequence, then different sequences must encode different traits. The four-letter alphabet of A, T, G, and C somehow spelled out the instructions for building proteins, which in turn determined an organism's characteristics. This insight connected molecular structure to classical genetics in ways that had never been possible before. Gregor Mendel's inheritance patterns, discovered in the 1860s through experiments with pea plants, could now be understood in molecular terms. Dominant and recessive traits reflected different versions of DNA sequences. Genetic linkage occurred when sequences were located close together on the same chromosome. The double helix also explained how genetic diversity arose within species. During sexual reproduction, chromosomes from two parents combined, mixing their DNA sequences in new combinations. Additionally, occasional replication errors introduced novel sequences that natural selection could test. This molecular understanding of heredity and variation provided the missing link between genetics and evolution. Watson and Crick's paper, published in Nature magazine on April 25, 1953, was remarkably brief, just over 900 words, yet it transformed biology forever. Biology. The final sentence captured the model's revolutionary implication with characteristic British understatement. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This modest statement concealed one of science's greatest insights. The double helix hadn't just revealed DNA structure, it had illuminated the fundamental mechanism by which life perpetuates itself across generations, maintaining continuity while enabling change. Behind the triumph of the double helix discovery lay a complex web of personalities, rivalries, and ethical questions that would shadow the achievement for decades. While Watson and Crick received immediate acclaim for their model, the contributions of other scientists, particularly Rosalind Franklin, remained largely hidden from public view. Franklin's role in the discovery was both crucial and controversial. Her X-ray crystallography work had provided the key experimental evidence that made Watson and Crick's model possible. Photo 51 revealed DNA's helical structure. Her precise measurements determined the helix's dimensions. Her analysis showed that phosphate groups were located on the outside of the molecule. Without this data, the Cambridge duo could never have built their accurate model, yet Franklin remained unaware that her experimental results were being shared with competitors. The revelation came through a chain of communication that raised serious questions about scientific ethics. Maurice Wilkins had shown photo 51 to Watson during a chance encounter. Additionally, Franklin's detailed measurements had been shared through an internal Medical Research Council report that Watson and Crick could access as council-funded researchers. This data sharing occurred without Franklin's knowledge or consent. In the competitive atmosphere of 1950s science, such practices weren't uncommon, but they violated modern standards of research collaboration. Franklin was essentially competing in a race where other participants had access to her data while she remained unaware of their progress. The gender dynamics of the era added another layer of complexity. Franklin worked in a scientific environment where women faced significant barriers to advancement. She was excluded from the senior common room at King's College, where male colleagues gathered to discuss research informally. Her authoritative manner and uncompromising standards sometimes created friction with male colleagues who weren't accustomed to women as intellectual equals. Franklin's relationship with Maurice Wilkins was particularly strained. Both brilliant scientists, they had fundamentally different approaches to research collaboration. Wilkins expected Franklin to work as his assistant on the DNA project, while Franklin viewed herself as an independent researcher with her own scientific agenda. These conflicting expectations created tension that hindered their potential collaboration. The personal dynamics between Watson, Crick, and Franklin were equally complex. Watson later described Franklin in unflattering terms in his memoir, The Double Helix, portraying her as difficult and unattractive. These characterizations reflected the casual sexism of the era and obscured Franklin's genuine scientific brilliance. Crick, by contrast, maintained respectful relationships with Franklin and recognized her contributions more fully. Franklin's scientific approach also differed fundamentally from Watson and Crick's methodology. Where the Cambridge pair relied on model building and theoretical insights, Franklin insisted on accumulating overwhelming experimental evidence before drawing conclusions. Her caution was scientifically sound, but cost her priority in the discovery race. She was preparing to publish her own structural model when Watson and Crick announced their breakthrough. The timing proved tragic for Franklin's recognition. She died of ovarian cancer in 1958, just 4 years after the double helix discovery. When Watson, Crick, and Wilkins received the Nobel Prize in Physiology or Medicine in 1962, Franklin couldn't be honored alongside them. Nobel Prizes aren't awarded posthumously. This exclusion has generated ongoing debate about scientific recognition and gender bias. Franklin's X-ray crystallography work was technically superior to most contemporary research, and her experimental insights were essential to solving DNA's structure. Yet her contributions remained largely invisible to the public for decades after the discovery. The story also highlights how scientific breakthroughs emerge from complex collaborations, competitions, and conflicts. The double helix wasn't discovered by isolated geniuses working alone. It resulted from interconnected research efforts across multiple laboratories. Franklin's experimental precision, Watson and Crick's theoretical insights, Wilkins' structural analyses, and Chargaff's chemical discoveries all contributed to the final breakthrough. Understanding these human dimensions doesn't diminish the scientific achievement, but rather illuminates how knowledge advances through both cooperation and competition, individual brilliance and collective effort, triumph and tragedy intertwined in the eternal quest to understand life's deepest mysteries. The publication of Watson and Crick's double helix model in April 1953 marked the beginning of a scientific revolution that continues to this day. Within hours of the paper's appearance in Nature magazine, researchers worldwide recognized that biology had been fundamentally transformed. The discovery didn't just solve DNA's structure, it launched the molecular age of life sciences. The immediate impact was felt across multiple scientific disciplines. Biochemists began investigating how DNA replication actually occurred in living cells, leading to the discovery of enzymes like DNA polymerase that copy genetic material with extraordinary precision. Geneticists could finally understand how mutations arose and how they affected organism traits. Evolutionary biologists gained molecular tools to trace relationships between species, and understand how life diversified over millions of years. Within a decade, the double helix discovery had spawned entirely new fields of research. Molecular biology emerged as scientists learned to manipulate DNA directly, cutting and splicing genetic material with unprecedented precision. The genetic code was cracked by 1966, revealing how DNA's four-letter alphabet spelled out instructions for building proteins using 20 different amino acids. The medical implications proved revolutionary. Understanding DNA's structure enabled researchers to identify genetic diseases at the molecular level. Sickle cell anemia was traced to a single base change in the hemoglobin gene. Huntington's disease, cystic fibrosis, and thousands of other inherited disorders could be understood in terms of specific DNA sequence alterations. This knowledge opened pathways to genetic testing, counseling, and eventually gene therapy. The 1970s brought recombinant DNA technology, allowing scientists to insert genes from one organism into another. This breakthrough enabled the production of human insulin in bacteria, growth hormone in yeast, and countless other medical products in microbial factories. The biotechnology industry was born, transforming medicine from a practice based on observation and tradition to one grounded in molecular understanding. By the 1980s, DNA fingerprinting revolutionized forensic science and paternity testing. The unique sequence variations in each person's genome provided definitive identification methods that transformed criminal justice systems worldwide. Cold cases could be solved decades after the crimes occurred, and wrongly convicted individuals could be exonerated through genetic evidence. The 1990s launched the Human Genome Project, an international effort to sequence all 3 billion base pairs in human DNA. This 13-year endeavor, completed in 2003, exactly 50 years after the double helix discovery, provided humanity's first complete genetic blueprint. The project cost $3 billion and involved thousands of scientists across 6 countries, representing one of the largest scientific collaborations in history. The genomic revolution accelerated dramatically in the 21st century. DNA sequencing costs dropped by a factor of 10 million between 2000 and 2020, making personalized medicine possible. Today, doctors can analyze a patient's genetic profile to predict disease risks, select optimal medications, and tailor treatments to individual genetic variations. CRISPR gene editing, developed in the 2010s, provided unprecedented precision in modifying DNA sequences. This technology enables researchers to correct genetic defects, enhance crop yields, and potentially eliminate hereditary diseases. The possibilities seem limitless, though they raise profound ethical questions about human genetic enhancement and the boundaries of medical intervention. The double helix discovery also transformed our understanding of life itself. DNA analysis revealed that all living organisms share common ancestry, with genetic similarities reflecting evolutionary relationships. Humans share 99% of their DNA with chimpanzees, 50% with banana plants, and significant portions with bacteria. This molecular unity of life confirmed Charles Darwin's insights about common descent, while providing unprecedented detail about evolutionary history. Looking forward, the double helix revolution continues accelerating. Artificial intelligence analyzes vast genomic databases to identify disease patterns and drug targets. Synthetic biology engineers organisms with novel capabilities. Space agencies study extremophile bacteria to understand life's limits and possibilities beyond Earth. From Watson and Crick's elegant model to today's genomic medicine, the double helix discovery represents science at its most transformative. It revealed life's fundamental mechanism, while opening infinite possibilities for human knowledge and capability. The revolution that began in a Cambridge laboratory 70 years ago continues reshaping our world, promising solutions to challenges we're only beginning to imagine. The secret of life, once hidden in molecular shadows, now illuminates pathways to humanity's future.